Is that a viable alternative?
It's a really important question.
Yeah.
Biological control basically uses natural enemies, could be beneficial bacteria, other fungi, to fight the specific pests or pathogens you're targeting. And one potential advantage is that developing these is often much cheaper than creating a brand new chemical product from scratch.
Okay, cheaper sounds good. Can you give us some examples? What kind of BCAs are being looked at for mushrooms?
Sure. One promising group involves Bacillus species. These are types of bacteria, gram-positive bacteria, that work in some pretty cool ways. Think of them as tiny bodyguards for your mushrooms. They produce natural antimicrobial compounds, things like cyclic lipopeptides, that essentially poke holes in fungal cells or stop spores from germinating. They also release enzymes that can break down fungi, plus they compete with the bad guys for space and nutrients. Some can even form protective biofilms on the mushroom tissue itself.
Like a multi-pronged natural defense system. Are there specific Bacillus strains showing promise in the lab?
Yes, there are. For instance, Bacillus billizensis QST713. You might know it. It's in a commercial product called Serenade ASO. This one has shown activity against green mold trichoderma. And in lab tests in vitro, as we say, it also inhibited the fungi causing dry bubble and cobweb. There's another one, Bacillus billizensis cos, a newer isolate. It also showed good inhibition of dry bubble and cobweb in the lab. The liquid it grows in, its culture filtrate, contains antifungal compounds, including a protein called sapillicin.
Okay, so they look good in the lab. Now, here's what I find fascinating. Often these BCAs show really strong results in vitro, but when you try them out on a larger scale in actual crop trials in vivo, the effectiveness sometimes seems, well, limited compared to the traditional fungicides. What's happening there?
That is the million-dollar question, isn't it?